Computational biomedicine pillar of the bioengineering department at George Mason University is located in Peterson Hall in the Fairfax campus and comprises the Laboratory for Computational Hemodynamics, directed by Professor Juan Zebral, the Laboratory for Signal and Video Analytics, directed by Professor Vicky Economidou, and the Laboratory for Data-Driven Biomechanical Simulation, directed by Professor Chi Wei. Researchers in the computational biomedicine pillar focus on the development and application of image-based, patient-specific computational modeling and simulation, as well as data analytics to study a variety of diseases, surgical interventions, and medical devices. The goal of this research is to contribute to the basic understanding of different diseases, enhance diagnostics by complementing medical imaging with modeling and simulations, improve treatment planning and personalized therapies, and evaluating and optimizing devices and surgical procedures. The Laboratory for Computational Hemodynamics specializes in blood flow simulation in humans. The lab is directed by Professor Juan Zebral, who obtained his PhD from George Mason University in 1996. His main areas of expertise are in the construction of patient-specific computational models of brain arteries and aneurysms from three-dimensional images, and using these models to study cerebrovascular diseases and their endovascular and surgical treatment. The lab is currently working to understand the mechanisms responsible for the local weakening of the vascular walls that lead to the enlargement and eventual rupture of intracranial aneurysms, creating rupture probability models to assess rupture risk of cerebral aneurysms and to evaluate and optimize endovascular devices such as flow-diverting stents. An example project focuses on understanding the different failure modes of cerebral aneurysms and the conditions that predispose aneurysms for destabilization and eventual rupture. For this purpose, we are collaborating with researchers from the University of Pittsburgh and neurosurgeons from 4 clinical centers to collect tissue samples, medical images, clinical information, and combine this data with ex vivo multiphoton microscopy, mechanical testing, and image-based patient-specific computational models to understand the interaction between blood flows and the biological responses of the wall that lead to wall degeneration and weakening. The ultimate goal of this research is to improve patient management by selecting only high-risk aneurysms for surgery. This research is funded through a multi-center NIH grant. In a second project, we are collaborating with clinicians from the Mayo Clinic and a major clinic in Buenos Aires, Argentina, to assess the likelihood of successful treatment of complex cerebral aneurysms using flow-diverting devices. For this purpose, we build patient-specific computational models of the aneurysms and perform virtual interventions to predict the long-term outcomes of the procedures. The goal of this research is to personalize the treatment by selecting the most appropriate device or devices for a given aneurysm to maximize the chances of a complete and stable aneurysm occlusion. This project is also supported by the NIH. Examples of students' projects include, first, studying blebs or secondary outpouchings as potential biomarkers for aneurysm rupture risk, which is the focus of Ms. Setara Salimi, a PhD student in the Computational Hemodynamics lab. Second, understanding the mechanisms of aneurysm wall enhancement in magnetic resonance imaging and its potential as an indicator of wall vulnerability, which has been carried out by Ms. Sarah Haddad, another PhD student in the lab. And finally, investigating the potential negative effects of inserting catheters to treat ischemic strokes caused by the occlusion of a cerebral artery, which has been conducted by Ms. Medhini Solsal, an undergrad student in the bioengineering program. In addition to high-end graphic workstations in the different labs, computational equipment in the biomedicine pillar includes a number of data servers and supercomputing clusters which are housed in the Acquia data center in the Fairfax campus. These computers are used to carry out high-performance parallel computations and simulations and to store and analyze large amounts of data. If you are interested in learning more about us, our research, or our programs, please visit the Bioengineering Department website at bioengineering.gmu.edu. Thank you very much.